So this video is going to cover T cell development. T cells, like all immune cells, begin their life in the bone marrow as pluripotent hematopoietic stem cells, right? And when those cells undergo division, they self-renew, but one of their progeny, we'll call that a progenitor cell, it's of the lymphoid lineage, and it's going to eventually turn into a large population of T cells. But this isn't going to happen in the bone marrow, right? These cells actually leave the bone marrow and enter an organ called the thymus. So if you remember, the bone marrow and the thymus are both considered primary lymphoid organs because those are locations where immune cells develop. In the bone marrow, the B cells complete their development. In the thymus, T cells development. So we call it a primary lymphoid organ. And it's found in the upper anterior thorax, just above the heart, right? So right in your chest. That's where your thymus is. Right? And what are you going to find in your thymus? Well, you find these very interesting epithelial cells called thymic stromal cells. And these cells are going to send signals to these cells arriving to tell them to start T cell development and going to test them so that when they leave out, when the cells leave the thymus, they are functional working T cells. So when the cells arrive here, they get signals to start to go toward the T cell lineage. And so these cells are now called immature T cells, or thymocytes. So thymocyte sounds like a cell that you find in the thymus, which is true, but the thymocytes are actually immature T cells, right? The cells that actually make up the thymus that stay in the thymus are these thymic stromal cells, which are epithelial in nature. But thymocytes, they enter the thymus and hopefully they leave the thymus nice immature T cells, right? And so that's what we're looking to get. We're looking for T cells that will leave and have a nice functional T cell receptor that will wander your body, checking your MHCs to see if there are any peptides there that are recognized and therefore are non-self peptides. And the T cells we're looking for are CD8 positive and CD4 positive T cells, right? So there are really three things that occur in the thymus. We're going to talk about the first in this video, and then there will be two more videos on the other. There's T cell receptor gene rearrangement, and we're going to focus on the alpha chain and the beta chain. Your book in Chapter 7 talks about gamma and delta T cell receptor. Don't worry about any of that. It's interesting stuff, but we're not going to have time to get into that. You could look in your textbook if you want to know more about those cells. But we're going to focus on the alpha-beta chains of the T cell receptor and gene rearrangement. After that occurs, positive selection occurs, and then negative selections occurs. And those are important for training T cells to check your MHCs and to ignore your peptides. And we'll see that in later videos. Okay. And like I just mentioned here, the purpose of all this is to make sure that your T cell receptors that you rearrange will check your MHCs, right? If it's a CD8 cell, it'll check your MHC1s. If it's a CD4 cell, it'll check your MHC2s. But you want the cells not to activate if those MHCs are carrying self-peptides. So that's really the purpose of what's going to happen in the thymus. All right. So let's start out with these thymocytes, right? And so these cells, they've arrived in the thymus, these progenitor cells, and they're going to start to undergo T cell development. So we can call them immature T cells, or we'll call them thymocytes. In your book, they're typically referred to thymocytes. And the first thing that's going to happen is they're going to rearrange the beta chain of the T cell receptor. And so you recall from Chapter 5 and even from B cell development that all these cells that have arrived here that are going to undergo T cell development, their genes, the alpha gene and the beta gene, right, they have lots of gene segments, variable, diversity, joining, and they all need to be cut and pasted together before they can make a functional open reading frame that can make a protein. Right? So we have a special name for these cells that are haven't undergone or are undergoing gene rearrangement, specifically of the beta gene. These cells are called double negative thymocytes. And why do we call them double negative? It's because of the absence of the CD8 and the CD4 proteins on their surface. There are lots of proteins on the surface of thymocytes, but the proteins that are absent are CD8 and CD4. Those don't get turned on until later. So if I looked in your body right now and I pulled out a bunch of cells, T cells, and they were CD8 minus and CD4 minus, I would know exactly where those cells came from. They came from your thymus. They were undergoing gene rearrangement of the beta gene. So those are double negative thymocytes. They just give us an indication of where the T cells are in their development. Right? So in these double negative cells, the genes that turn on are RAG. If you remember that recombination activating gene that's involved in cutting and pasting gene segments together, landing on the RSSs randomly, right? Picking a V and a D and a J, linking them together to hopefully make a functional open reading frame that will make a protein. And the enzyme TDT, terminal deoxynucleotide transferase, which puts N nucleotides between the joints, and you also have P nucleotides as well there. So this is just like gene rearrangement of the heavy and light chains. So hopefully, if this all goes successfully, and if it doesn't, the RAG can pick other V's and other D's and do a little more shopping around to make sure that when these gene segments are put together, it's a functional protein. It makes a functional protein. So the DNA is read with an ATG, it reads all the way through the mRNA. The protein makes a variable chain, a variable region, and a constant region. And now we've made, great, a fully functional TCR beta chain. So are we done? No, we still need to do the alpha chain, but we need to prove to the T cell that we can live, that we should live and survive. So we need to assemble some sort of pre-receptor to just prove to the cell that we've done this successfully. And so this is very similar to B cells. In these T cells, they make a pre-T cell receptor. So they assemble all the proteins that are normally found in a T cell receptor, the CD3 proteins, the zeta proteins. We now have a good beta protein. We don't have an alpha protein yet because we haven't rearranged the alpha gene, but we do have a placeholder protein or a surrogate protein for the alpha chain called PT alpha. Right? So PT alpha is the stand-in for the TCR alpha. We haven't done that yet. We don't want to spend any energy on that until we get the beta chain fully made. And now the beta chain's made, and we're great. So of the cells that start down this path, about 80 of them go through and finish this process, which we call a productive rearrangement. If it's unproductive, that cell just dies, and we go and we do it again to other cells. So now we've got a pre-T cell receptor, right, and we've got a good beta chain. Right? We're going to call these cells pre-T cells. Right? Before they were immature T cells or immature thymocytes. These are pre-T cells. So what is going to happen to a pre-T cell? First thing, it's going to undergo mitosis. We want to make more of these cells so that we can send their progenitors down paths to do alpha chain gene rearrangement. So that's one thing that's going to happen. The other thing that's going to happen is that we can turn on the genes that make those proteins, CD4 and CD8. So now CD4 and CD8 go to the cell surface. I don't know if I mentioned this, but the pre-T cell receptor actually stays in the endoplasmic reticulum. It doesn't actually go to the cell surface, just like the heavy chain, the pre-B cell receptor. But the CD4 and the CD8 proteins, those are now turned on, and so they go to the cell surface, and now we can call these T cells double positive thymocytes. So your pre-T cells are CD8 positive and CD4 positive, right? Whereas the immature thymocytes that have not finished B cell gene rearrangements, we refer to those as double negative thymocytes. So you should need to keep track of when T cells have their CD4 and CD8 off, when they turn them on, and now we're going to see eventually one of those will go back to being turned off. So keep track of those. So let's say we have a bunch of pre-T cells, and they're all double positive, right? CD4 and CD8, that one cell that we rearranged underwent mitosis and mitosis. We made maybe 100 cells out of this one. And so they all have the identical beta chain, and I just picked a V2, D1, J, V2, V10, D2, J1. That's what I sort of randomly picked for this T cell. So it's got a beta chain with a variable region, so I drew a funky little shape due to the VDJ recombination, and the P nucleotides and the N nucleotides gives that variable region some unique shape. I also drew CD4 and CD8 on all the cells because they're all double positive. Now what happens to these double positive cells? Well, they're going to undergo gene rearrangement in the alpha gene of the T cell receptor. So they're variable gene segments, they're joining gene segments, RAGs turn back on to cut and paste, right? And so that top cell, let's say that top cell picks V17J50, puts in some random nucleotides at TDT, and so you make a alpha chain protein, right? And its variable region has some really interesting shape because of the DNA sequences in the V and the J and the N nucleotides and the P nucleotides. This other cell, well, it picked V11J18, right? So its DNA sequence is going to be different than the cell on the top. So its variable region of the alpha change protein, different shape, right? Cell at the bottom, it picks a different V and a different J, 66, 42, right? So when it makes a protein, the amino acid sequence different than the other one. So its alpha chain, variable region, has a different shaped peptide binding domain. Right? So now what are we left with? Well, we're left with cells that have functional T cell receptors. They've made their proteins, they sent them to the surface. Now, at this point, we need to know, do these T cell receptors, do they interact with peptide? Do they interact with self-MHCs? Because we don't know that at this point. They just have these variable protein domains at the end. We don't even know if these work. And in fact, as we'll learn, most of these cells are going to die because they are actually, those shapes that you made, they're not very good. So this cell is going to have to undergo certain checks to make sure that we're going to release it into the body, and it's actually going to be worthwhile to have these cells. And that the next video is going to talk about positive selection, which is the next thing that these cells are going to undergo. Right? But we have at least finished T cell receptor production, and so all of these T cells have a T cell receptor on their surface. They have this funky looking shape which may interact with the peptide, which may not interact with the peptide. It may interact with a self-peptide, and if it does, we're just going to get rid of that cell. It may interact with a viral peptide, which is great, but we don't know that because we don't know about the virus yet, right? These cells have never seen viruses, they've never seen pathogens, they don't know anything. So they'll have to go a little bit of training before we let them out into the world.